All right, so today we're doing something a little different. We're not going to be digging into some dense research paper or a breaking news article. Nope. Instead, we're going to uncover some of the most amazing stories about life itself, and we're going to find them in the last place you'd ever think to look, a biology textbook problem set. Seriously. So let's dive in.
I mean, think about it. What if the answers to some of biology's biggest, most fundamental puzzles, things like scale, information, sheer speed, what if they were just hiding in plain sight, just waiting for someone to, well, to do the math? That's exactly what we're going to do. We're going to solve these problems, and as we do, we'll start to see the absolutely incredible engineering that makes all life, including us, possible.
Okay, first up on the docket, let's try to wrap our heads around the sheer density of life. A single cell isn't just some empty bag of water. Oh no, it's way more like a jam-packed, bustling city, just humming with molecular machinery. But the real question is, just how crowded is it in there?
So let's just try to get a sense of the scale here. Take a typical muscle cell, maybe 50 micrometers across. I mean, it's tiny, right? But if you could, just theoretically, fill that cell with only one type of protein, actin, the stuff that makes muscles contract, you could cram in 65 billion of them. 65 billion. That is a density that's it's almost impossible to imagine. And it's not just tiny little proteins either. What if we look at something bigger? In a liver cell of about the same size, you could fit roughly 37,000 mitochondria, the powerhouses of the cell. So this tells us something really important. Cells aren't just packed, they are packed to the gills with these huge complex structures, and every single one has a job to do. And believe it or not, this whole principle holds true even for the smallest of the small. Let's take a tiny E. coli bacterium. The machinery it uses to build proteins, all 15,000 of its ribosomes, they take up about 4% of the cell's entire volume. And its protective outer shell, that's another 5%. So right off the bat, we see this is not empty space. It's an incredibly crowded environment where every last bit of real estate matters. All right, so we've established that cells are basically packed with stuff. But what about maybe the most important component of all? What about the instructions, the blueprint? Let's take a look at how a cell manages to store its entire genetic code. And this right here, brings us to a paradox that is just stunning. An E. coli cell, it's only about 2 micrometers long. But if you were to take its DNA molecule, it's a single circle, and stretch it all the way out, it would be about 1,600 micrometers long. That's almost 1.5 millimeters. You could see that with your naked eye. So how in the world does that work? I mean, how do you cram a piece of thread that's 800 times longer than the box it's supposed to fit in into that box? And not just cram it in, but do it without it becoming a completely tangled, useless mess. It really does seem impossible, right? Well, the answer is just one of biology's most beautiful and elegant solutions. It's called supercoiling. The DNA is twisted, it's folded, and it's coiled back on itself with just incredible precision. It gets compacted down into this tiny, perfectly manageable bundle called a nucleoid. It's nothing short of a masterclass in organization and information storage. Okay, so we've talked about scale and we've talked about information. Now, let's switch gears and look at something that's maybe a little more subtle, but trust me, it's every bit as critical. Let's talk about shape. Because in biology, the three-dimensional structure of a molecule, well, it's pretty much everything. So let's start with a classic question you might have even wondered about yourself. You've got vitamin C from, say, a rose hip, and you've got synthetic vitamin C made in a lab. If the chemical formula is identical, does it matter where it came from? Is one better? Well, the simple answer is no. If they're both pure L-ascorbic acid, they are the exact same molecule. End of story. But this simple question actually opens the door to a much deeper and way more important concept in biology. And that concept is something called stereoisomers. Now, the easiest way to think about this is to just look at your hands. Go ahead, hold them up. They're mirror images of each other, right? But you can't perfectly lay one on top of the other. Your thumbs will be pointing in opposite directions. A ton of biological molecules are just like this. They have a left-handed and a right-handed version. We call them enantiomers. They're made of the exact same parts, but their 3D arrangement is a mirror image. Okay, so why does this matter? Well, this handedness has absolutely huge implications, especially in medicine. Take this molecule, isoproterenol. It's a drug used for asthma.
Now get this, its right-handed version is 50 to 80 times more effective at opening up your airways than its left-handed mirror image. We're talking about the exact same atoms, the same bonds, but a completely different effect in the body. So here's the takeaway, the really crucial point. Your body's receptors are basically like tiny locks, and molecules are the keys. A key has to have the exact right shape to fit into the lock and open it. A key that's a mirror image of the correct one, even if it's made of the exact same stuff, it just won't work. It won't open the lock. And that's how your body can tell the difference between these two-handed molecules.
And this isn't just a lab curiosity. It has real-world consequences. For example, years ago, there were two drugs, dexedrine and benzedrine, chemically identical. But the recommended dose for benzedrine was double the dose of dexedrine. So what gives? Well, it turns out dexedrine was the pure, active, right-handed molecule. Benzedrine, on the other hand, was just a 50-50 mix of the active one and the useless, inactive, left-handed one. So you literally needed to take twice as much to get the same therapeutic effect. Like I said, shape is everything.
All right, for our final puzzle from this problem set, we're going to talk about speed. Because life, at its core, operates on some very, very strict deadlines. If you're too slow, you don't eat, or worse, you get eaten. So how has biology solved this fundamental challenge of moving things around quickly and efficiently?
Well, it all starts with a pretty simple principle from geometry, actually, the surface area to volume ratio. Just look at this comparison. A tiny little bacterium has a huge amount of surface area compared to the tiny volume inside of it. But a big amoeba, it's the opposite. It has very little surface area for its massive volume.
So why on earth does this ratio matter? Well, because a higher ratio means you can exchange things with your environment way, way faster. Nutrients get in, waste gets out, lickety-split. This is what fuels an incredibly high metabolic rate. It's the reason some bacteria can double their population every 20 minutes, while our much, much larger cells take a full 24 hours to do the same.
Okay, so that explains speed for single cells. But what about inside a big organism like us? How does a signal get from your spine all the way down to your toes? We're talking about a journey of over a meter. Just letting things randomly float there, simple diffusion, that is definitely not going to cut it. So how does the body solve this long-distance transport problem?
Well, let's run the numbers one more time. Our neurons, our nerve cells, they use these little packages called vesicles that travel along these internal highway tracks. Now, if one of those vesicles is moving at about 1 micrometer per second, to travel 2 meters from your spine to your toes would take about 23 days. Yeah, 23 days for one single package to get delivered.
Now, I know what you're thinking. 23 days sounds painfully slow. But here's the thing. Compared to the months, maybe even years it would take for that same package to just randomly diffuse its way there, that 23-day trip is a biological superhighway. And that's the whole point. Life had to evolve these directed, active transport systems. Without these internal highways, sending signals and nutrients over long distances would be flat out impossible. And complex organisms like you and me, we just couldn't exist.
So there you have it. Just by working through a few math problems from a textbook, we've uncovered these huge, profound principles that govern all life as we know it. We've seen the incredible density inside a cell, the absolute genius of information storage, why a molecule's shape is everything, and how life evolved to conquer speed and distance. And it all leaves us with one final, really fascinating question that was posed in the problems. We know that carbon has solved all these challenges with incredible elegance, but could another element, something like silicon for instance, ever have done the job? Could it have formed the basis for life? Now that is a thought to chew on.